Good day. This is the Professor, CBMD PhD, and today's Sunday, April 16, 2023. It is 4:04 and 24 seconds, a.m. Eastern Daylight Time, and today's research, T cells. So let's hop to it here. Has two different regulatory T cell populations. Study findings could lead to new paradigm shift in how autoimmune diseases are treated, allowing for suppression of autoantibodies while maintaining immune protection. Date April 7, 2023. Source: Children's Hospital of Philadelphia. When it comes to autoimmunity, the prevailing wisdom has been that the only way to stop inflammation is to suppress the immune system broadly, making patients more susceptible to infection. However, that is only true if all T cells come from the same place. What this study shows is that there are two different T cell lineages, which means you might be able to have your cake and eat it too, suppressing inflammation due to the autoimmunity while allowing T cells that fight infection to thrive. Germinal centers are spherical collections of cells inside tonsils, lymph nodes, and the spleen that orchestrate interactions between T follicular helper T cells and B cells. Although the proper function of T follicular cells is likely important to immunological health and their dysfunction a potential contributor to various disease states, few studies have assessed the biological roles of human T follicular cells, and none have addressed where they come from or how they develop within tissues. And to solve this problem, the researchers used a computational in vitro and in vivo techniques to describe the origins, functions, and positions of T follicular cells within germinal centers. As since germinal centers are located in secondary lymphoid tissues, the lymphoid nodes, spleens, and tonsils, the researchers analyzed tonsils that had been removed from healthy donor patients. Using an interlocking suite of single-cell technologies, the researchers were able to show that there is one subpopulation of T follicular cells that is induced by T follicular helper cells, which they call IT follicular cells, and another subpopulation that were naturally derived from Treg, a subpopulation of T cells that are responsible for moderating the immune system, which they call NT follicular cells. In doing so, this demonstrated that there are two developmental trajectories, Treg to NT follicular cells, and T follicular helper cells to IT follicular cells. Once the researchers identified these two subpopulations of T follicular cells, they analyzed whether these two regulatory T cells express a surface protein CD38 differently. They found that IT follicular cells express CD38, whereas NT follicular cells do not. They were also able to catalog the precise location of these different subpopulations within the germinal centers, in addition to demonstrating their developmental path and ability to support B cell function. And this raises a question of whether we can selectively deplete IT follicular cells through anti-CD38 treatments while leaving NT follicular cells intact. A similar approach could also potentially be used in a therapeutic context to boost immunity in patients with weakened immune systems. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.